This is the recipe for soft French bread. I will be following the procedure in this recipe to produce the bread, as well as talk about the chemistry that is going on while I am making it. You can pause the video and read through the procedure and see the ingredients before you watch the video of me making the bread and talking about the chemistry behind it. This is the chemistry of bread making. First we have our ingredients: water, flour, sugar, active dry yeast, oil, and salt, and also a big bowl to combine it all. So first we're going to add warm water. Next we'll add sugar and let the sugar dissolve in it. And we are also adding yeast. Then we will mix those with a whisk. While the yeast and sugar are mixing, yeast are single-celled fungi that help convert sugar into carbon dioxide, and those bubbles that you see forming and the foam are carbon dioxide, which will help the bread to rise. Next, we add salt. Salt not only adds flavor to the bread, but also slows dough fermentation and strengthens the gluten structure to make the dough more elastic. Next, I added oil. Oil is used as a source of fats, which will weaken the gluten network, resulting in a softer bread. The fats coat some of the proteins that form gluten and will prevent them from forming hydrogen or disulfide bonds, overall weakening the gluten network and softening the bread. Fats also stabilize the carbon dioxide gas bubbles, increasing the loaf volume. Next, we will add flour. Flour contains a high level of glutenin and gliadin proteins. These classes of proteins are large molecules built of numerous amino acids and are collectively referred to as gluten. When flour is added to water, the gluten proteins are able to line up with each other and interact. They can form hydrogen bonds and disulfide cross bridges between their chains, eventually forming a large gluten network throughout the dough. Flour also contains starch, which is composed of long chains of sugar molecules. The enzyme amylase in the flour converts the starch to maltose, which is then converted into glucose by maltase in the yeast. Along with other sugars, glucose can be used by the yeast for fermentation. All of the ingredients are mixed to start creating the dough. Once the flour is mixed and starts sticking together and doesn't stick to my hands anymore, we will let it rise and ferment for 10 minutes. Kneading the dough helps the gluten proteins to uncoil and interact with each other more strongly and strengthen the network. Kneading also ensures that there are a large number of carbon dioxide bubbles present in the dough in order to expel the gas before baking. The dough is left to ferment in 10-minute increments 5 times and kneaded in between each in order to help the proteins interact, as well as let the dough rise and start fermentation. As you can see, the more that the dough is kneaded, the less sticky it becomes, and the more it becomes one giant ball of dough instead of a sticky mess. So after the dough was kneaded and allowed to rise for 5-10 minute intervals, you can start shaping it into the loaf of the bread. The white powder on the bottom of the baking sheet is cornmeal, which will allow the bread not to stick to the pan, as well as help darken it during baking. The bread was then left to ferment for 30 minutes. During fermentation, the yeast will metabolize glucose to produce carbon dioxide and ethanol. Carbon dioxide helps the bread rise, and ethanol is boiled off during the baking process. Not all of the sugar is metabolized by the yeast and will actually participate in the Maillard reaction. During baking, there are a series of reactions called Maillard reactions between sugars and amino acids that occur rapidly above 140 degrees Celsius. These reactions produce a large range of products that can add flavor to the bread and also help form the bread's crust. The carbonyl group of the sugar reacts with the amino group of the amino acid, producing N-substituted glycosylamine and water. The unstable glycosylamine undergoes Amadori rearrangement, forming ketosamines. The ketosamines can react further and produce brown nitrogenous polymers and malanoidins that result in the brown color of cooked crust. That is why the bread turns brown during and after baking. And this is the final finished bread product. Thank you for listening to the chemistry of bread making.